I'm Edward Warfield, the publisher of Citybiz, and I'm honored today to interview Jeff Galvin, who is currently the CEO of American Gene Technologies and will be the CEO of the spin-out company Addimmune. Welcome, Jeff.
Thanks, Edwin. Great to be here.
At the beginning of June, American Gene Technologies launched a new company, Addimmune. Can you explain the rationale for the spin-off?
Sure. So American Gene Technologies is a platform company, and we have a diverse set of developments. Some of them are actually in a pipeline that we intend to eventually get into the clinic. But something that got in the lead was our HIV cure project. And we just finished up a phase 1 on that and an analytic treatment interruption. And we were so impressed with the data that we felt that we would want to spin this out to give it additional focus and to maximize the chances of actually getting out there and commercializing a cure. Now, it's very important when I say stuff like that because, there's so many people that are desperate for a cure. And if they're listening to this interview, they need to understand that we have great phase 1 data and we have some really encouraging immunological observations and data out of the analytic treatment interruption that are encouraging, but there's still time to go. There's still development that needs to happen. We will roll into a phase 2, and we're going to start talking to the FDA about that shortly. But there's more clinical work that needs to be done, and this is going to take time. But the momentum is fantastic. So this is the encouraging part of it, is that we see that there is a good possibility of achieving a functional cure using gene and cell therapy, and that our approach seems to be on track. So, albeit that people still need to be patient, we're going to dedicate a huge number of people entirely to the HIV project. We're going to go out there and get additional funding, and we're going to drive this thing pedal to metal because we think that we've got line of sight on a very important innovation that people have been waiting for a long, long time.
And what is the status of the clinical milestones? Walk us through, bring us up to date on the actual milestones.
Sure. Well, of course, the primary endpoint of the phase 1 is safety. And out of the 7 patients that we treated, we had 0 serious adverse events. We had no adverse events that could be tracked back to the drug itself. And we recorded 100% of our secondary endpoints, which are called blood marker of efficacy. Blood markers of efficacy essentially refers to that the cells, this is an autologous cell therapy, that the cells that we infused into the patients engrafted, so they found a home there. They persisted, so the body did not reject them. They had no reaction against the body, so there were no adverse events because the cells were somehow attacking the body in some unusual way. And that they stuck around for as long as you would expect them to stick around without stimulation of the virus, because we kept all of the patients on their antiretrovirals for at least 180 days while we studied how the cells behaved. So the blood markers of efficacy were also 100%. Every single patient demonstrated that the cells behaved according to theory and consistent with how we believe they need to behave in order to fight HIV effectively in the bloodstream. So that was the phase 1 data, and we think that the data is good enough to justify a phase 2. That's why we're going to go ahead and apply for that. But also, we did what's called an IRB approved study that involved an antiretroviral withdrawal on the patients. So we had to re-enroll them, and we got approval from the IRB, which is essentially the committee that represents the hospitals or the doctors and makes sure that there's a second set of eyes on any experiments that might be done in that environment. And we also sent this withdrawal protocol to the FDA, and they remain silent, which is sort of their way of a tacit approval. It's not actually an endorsement, but at the same time, they're saying, we're not going to stop you. So we did get a chance to take 6 of the patients off of antiretrovirals, and all 6 of them showed an immune response that is characteristic of an effective fight against the HIV virus. And what do I mean by that? Well, normally you take somebody off their antiretrovirals that's well controlled, So they've got a very low viral load. You take them off, and what you see is their CD4 count starts to go down. And that's because the virus will attack the CD4 cells and start to deplete them out of the circulation system. So, but in the case of our patients, we saw those CD4 stable. Now, why is that? That's the whole intent of the project, is we are modifying CD4 cells so that they cannot be infected and depleted by HIV. And if they're not infected and depleted, if the HIV T cells are able to do their job, they should be able to clear HIV like you clear a cold or like you clear any other virus, right? So that we saw that the CD4 cells seem to be protected. The other thing we saw that's uncharacteristic of taking people off of antiretrovirals is normally you will not see a CD8 response, and that is because the CD4 cells being depleted out, they don't get a chance to call in the CD8s because they are the conductors of the immune orchestra, so to speak, the CD4s. And since they're not sending instructions to the CD8s don't respond to the virus. Well, in our case, we saw CD8s climb in all of the patients. So this was our first validation that we were seeing what might be an effective immune response. Well, then we thought, okay, let's bring these people back to zero virus again, because that first exposure to the virus is almost like a vaccination. It should prep those immune cells so that the second time they see the virus, they react more effectively. We got 4 of those 6 people to go off their antiretrovirals a second time, and all 4 of them showed a significant viral suppression. So that little auto vaccination, along with a second withdrawal, brought these patients down in a way that was exceedingly encouraging in terms of continuing on this path. So that's the clinical status so far. We have safety and we have blood markers of efficacy in the official phase 1. And then we have some really remarkable data showing that the T cells are responding as they should to HIV instead of as they normally do. Instead of being infected and depleted out, they're sticking around and they're mounting an effective fight against HIV. Now we just need to figure out how many of these do we need? Are there other things that we need to add to it to make it a functional cure? But we feel like we have line of sight on something that would get people off of antiretrovirals and maybe a one and done treatment that would render them immune to HIV for life.
So is the solution a cure or a treatment?
Well, the idea is it would be what's called a functional cure. Functional cure is different from a sterilizing cure. If you eradicate a virus totally from your body, like when you get over COVID, you have no COVID left in your body, most likely. Or I can't actually say that because I'm not a virologist, but some viruses can actually be completely eliminated from your body. But in a functional cure, you may still have some of the virus in your body, but the word functional means equivalent to cure. In other words, whatever the difference is makes no difference to your life. So in the case of HIV, we believe that the immune system, as modified by our cell therapy, is able to hold the virus down at levels where the person would not be contagious. So there would be no risk of them passing the virus. They could never develop AIDS because the viral level would be so low. And in fact, even if they got re-exposed to HIV, they couldn't recontract it because what would a tiny bit of HIV do in their body if their immune system could clean it up? So that's why we think it would give them permanent immunity. It would be a cure in that they would never have to take antiretrovirals again. They wouldn't need any further treatment. And it would be considered a functional cure because there might still be some HIV viral reservoir around, but any virus that were to erupt from that viral reservoir would make no difference in their lives.
Interesting. So can you share what's in the patent portfolio?
Sure. I mean, anybody can look it up because we have 25 granted patents in the United States and dozens of international patents. I think, and I can't verify this for a fact, but, this is the kind of feedback that I'm getting from our very well-educated patent attorneys that we're pretty much out patenting most companies in the gene and cell therapy area. That's a lot of patents, but what's exciting about our patents is that there are all these reusable components that could have utility in a wide variety of diseases, and that you can sort of mix and match these components to build a foundation of a new potential cure. So, what does that remind you of? Sort of like MS-DOS, right? You don't have to reinvent the wheel to write Microsoft Word or Microsoft Excel. And that's what we're doing. We have this platform of components. And in fact, they form the foundation on this HIV cure. You could think of that as our Microsoft Word off of this platform. And, it's our hopes that we keep on adding components to this platform over time, that we make it easier and more reliable and more efficient to create new gene and cell therapies. And that's the way that we hope to really have the maximum impact for patients. Because if we can enable other developers of gene and cell therapy products that could just go ahead and obtain our platform to reduce their costs by 90%, reduce their time by 90%, reduce their risk overall, right? You're going to get, a lot more attempts at cures for up to 10,000 diseases that I think eventually will fall to gene and cell therapy. So think of us as being in the early days of creating an iPhone, where curing a disease would be like writing an app on that iPhone, because the fundamental things that have to be done in almost anything that we're going to do with modifying your DNA in your body would be contained in the platform, and people wouldn't have to reinvent those components in order to do new innovations on top of it. So that's basically the patent portfolio. We have a lot of patents in HIV. We have that protected 6 ways to Sunday, because, of course, we have investors in the company, and if we cure HIV, we need to make sure that they get a reward. And so there's a bunch of patents that all come together. They're components. Some of them are components that could go into other chronic viral infection applications in the future, disease cures in the future. But, it basically forms a process and a molecule and, a modification of DNA that is a proprietary way of giving T cells a new function of being able to control HIV in the patient's body.
So who are the other key members of your team?
We have 50 key members of the team. I mean, because you just can't do it without, every aspect of a fully integrated, fully staffed company. So from the janitor up to the CEO, I like to feel that everybody is critical, and, and everybody in the company has stock. That's one of my fundamental beliefs. Now, we have some amazing folks with tremendous experience in developing drugs. For instance, Jeffrey Boyle, PhD, was actually former president of Addimmune in Frederick, and he wanted to get back into R&D, and he came on as CSO. But he's got over 20 years of drug development experience in FDA regulated projects in both drugs and medical devices. And so, we've got a great, foundational, person in him who can not just run R&D, but can direct an R&D organization to dot all the I's and cross all the T's that are necessary to get a drug out to the market. Another person that is really key in the organization would be Marcus Conan. He's our chief medical officer, and he has over 40 years of experience in HIV, having been on the front lines of the AIDS epidemic in San Francisco, and one of the first doctors to recognize a new virus was going around. He treated thousands of patients in the 80s and early 90s who passed away from AIDS. And then he was involved in clinical trials for the first effective treatments. So he's got a wealth of knowledge when it comes to clinical trials and HIV. and he runs our whole clinical ops program. So between the two of them, these guys are the ones that are doing the scientific and medical work. Then we have we have marketing and finance and business development. It takes money to make the company go, and we've raised almost $80 million over the last 10 years to go ahead and power this whole organization.
And so, are you working on a Series A for Addimmune?
Well, yeah, so Addimmune will be a new spin out, and it's going to take most of the employees from AGT with it, because we're going to put a big focus on that and try to head towards the public markets, which will involve, a round that we're doing right now. We're planning to raise $50 million. It's convertible debt. It's an institutional round. So it's like a Series A. And the plan is to give a preferred return to the early investors in that, and there'll be more details coming out about that soon.
And are you using AI in any capacity for? Is it impacting what you're working on?
So not really. AI will be indirectly impacting everything that we do, because if you look at what we do as a company, we sort of exist at the nexus of genetics, systems biology, and editing technologies. And what we look for are small changes that we can make in genetics that affect systems biology in a positive way for a healthcare outcome. So we're looking for things that we can improve in healthcare, things that we can cure with the existing editing technologies with all that knowledge. Now, that knowledge is benefiting from AI, big data, all of that stuff. And of course, we're resting on the computer revolution in terms of the robotics that we use in the lab and analysis of all our data. And there's big efficiencies that are brought by computers. And my previous, career was in computers in Silicon Valley. So yeah, AI is impacting us. We're using it around the margins, and that it's still useful when we're doing things like marketing things, text for the website or for press releases. It can be really helpful already. But I think that long term, AI will have a huge impact in developing drugs. And it's a matter of when we run out of low hanging fruit and when we think that AI will shine a light efficiently on new applications for our platform or new opportunities for components that we may, develop and put into that platform. So I would say that AI is not a big factor right now, but we're keeping an eye on it. And I certainly believe that in the future it will be big.
So, Jeff, is there any part of the Jeff Galvin story or the Addimmune story that you haven't shared with us that you'd like to share with us?
Well, I think that, the mission is what's important here, right? I got into this 15 years ago because I saw viral vectors for the first time, and I believed that what they were essentially were the diskette that would deliver software to the organic computer, the human cell. And I realized that if you can update the operating system of the cell, right, the DNA, you're now fiddling around with the root drivers of everything in your body. You can almost look at your body as being 37 trillion collaborating organic computers on a network that's connected electrically, chemically, and even physically, right? And that, now that we can get down to those root drivers, there's really nothing that can't be cured. And I think that that's an observation I made that made me realize that the mission of bringing that to the people, in comparison to any individual, including myself, is just huge. And so I've kind of lost myself in that mission. Now, my background just happened to be maybe appropriate to have that vision for the future of gene and cell therapy because I came out of computers, software, internet, IT. I was used to tech curves and technological revolutions. And quite frankly, even though that we call it biotech, I would say that gene and cell therapy is one of the first times that real tech, in terms of, sort of the tech curves, has come to biotech. And I think that what that means is that we're going to see a revolution in pharmaceuticals because there's a new drug development modality where every experiment relates to every other experiment, and things that you learn are reusable across many, many different things. And even this idea of the platform that we're developing is finally something where a platform brings efficiency and sort of directed development of drugs instead of random discovery or random generation of molecules that you, or monoclonal antibodies that you try out in the body, right? So this, sort of ability to design drugs now in a directed manner against specific things that we learn about genetics or about systems biology means that not only does it have great power to cure many diseases, but it's going to be like a snowball rolling downhill as well. That this massive knowledge that we're getting from the start of the Human Genome Project, right here in our backyard here in Rockville, right? What that did was it brought down the cost of sequencing and the synthesis, and it trained tons and tons of PhDs on, sort of the nexus of genetics and biology, and, made PCR, just abundant, and just set off a revolution or just a huge initiative in discovering more about the body with all these new tools in a way that, it's rare in any technology. But I think that lit the fuse on what I call DNA Valley, because I believe that within the DMV, and certainly within the United States, what we're going to see is a new, industry that Goldman Sachs says is a new $5 trillion industry. Well, that would be impressive on its own, given that pharmaceuticals is only $1.5 trillion per year, and Goldman Sachs thinks it's going to get driven to $5 trillion. That's big, but I think they grossly underestimated. I think that gene and cell therapy will cure 10,000 diseases. Then it'll be the fountain of youth, and then we'll ultimately control our evolution, because we're going to be able to design improvements into human beings. And don't be scared. You don't go run and watch Gattaca again right away. It's going to take 100 years to get to that point. But in the near term, it's going to be really beneficial, because we can talk cures instead of treatments. We can do one and done things that improve your life instead of treating you every day for a disease. And that's a bright future for everybody in this industry, but it's also a new thing that the United States will lead in globally. And why? Because it's really homegrown right here in the DMV. And, this is the epicenter of what I believe will be the gene and cell therapy revolution. I think that lots of great stuff is being done other places, but this place right here, Montgomery County, has the space to grow, and it has all the precipitants to grow something that looks a lot like the computer revolution in Silicon Valley. So that's why I call this place DNA Valley, and I'm hoping that that term will get some traction around here. I think it's an apt description.
It's nice being close to the FDA, right?
Close to the FDA, close to the NIH, and Maryland really leans forward in education. They have a lot of STEM graduates. So the workforce, is naturally evolved to the kind of work that we do. And I think it's a nice place to live as well. But look, the whole U.S. is going to participate in this, but I think you're going to see sort of a triangle from D.C. up through Frederick and Baltimore that may be something that grows very, very quickly because of some of the things that you just mentioned. There is an advantage to being in the shadow of the FDA, in the shadow of NIH. And it's especially good to be in an area where the earliest people who were thinking about this essentially were exploded out into the environment after 2 projects sequenced the human genome. Because remember, there were 2 sequencings of the human genome in this area that were in competition that happened just about simultaneously. And it was just an explosion of not just trained people, but dreamers who saw that this could really make a difference. And so there's a character around this valley, or this area, because it's not even a valley, but it reminds me so much of Silicon Valley from the days that I spent there in the San Francisco Bay Area, in the computer and software revolution. Well, I think that we have a possibility of creating a $20 trillion market globally. I think the U.S. is going to lead in this, and I think that Maryland is going to get more of their share. The DMV is going to be a place where this grows, just because the elements that we need are here. And we'll see. But remember that DNA Valley. idea, and let's talk again in a few years, and we'll see whether it's heading that direction. I think it will.
Well, Jeff, thank you for sharing your passion and vision. I enjoyed hearing it 6 years ago, and I'm excited about the latest chapter. And keep us apprised as you go beyond the DNA Valley to your next horizon. Thank you for your time.
Thanks, Edwin. Thanks so much for having me on your show.